Yeah.
Sorry,
Can you draw me?
a meme?
No,
No?
I saw something.
You might need to put in things that says.
Yeah.
it's recording.
Is the mouse at?
It's
Or
over
we can put
there.
it-
Oh, it's like, can I mirror this so I can see my presentation here as well?
Oh, you want to see it here?
Like, just in case I need to like,
Oh, thank you.
oh, thank
You want a clicker?
Well,
Then you still need to see it on here too. You can do both.
I mean, if I'm clicking out, so you can just like look at
Okay.
that, that's fine.
I just has to be plugged into one of the ports.
Yeah,
Yeah,
thank you, Brian.
no problem.
So this is the pointer and then.
The pointer, yeah, that works. On TV screens, it doesn't work anymore, right? The TV screens absorb it, but it still works on here.
But like, this is click forward and back.
It will go forward and backwards. And then that gives you, oh, because it's not hooked in yet.
Oh, the pointer works independently, okay.
So you need the USB in. And then you just gotta give the chance for the computer to catch up. And I think you should be good.
Okay, can you check?
Yeah.
Perfect. Okay,
Thank you.
well, I can make it mirror to you.
Wow, let's do it.
Yeah. I'm apparently a tech savvy, so. No.
That's, yeah.
Yeah. You're welcome. Can you try clicking it?
What happened there?
I have no idea. What? No. Please don't show my email. Show me my emails.
Fortunately enough, what happened?
It's now not loading.
Yes.
I think we have time. It's fine.
Like the computers.
Yeah.
What happened to PowerPoint?
No, it's not. It's on PDF.
Oh, that's why.
Trying to get. Why keep opening? Should be on.
Should we move it over to the second screen again?
Yeah. Like that.
Yeah.
Let's get going on this. First of all, I want to say we are in countdown mode for our poster session tomorrow afternoon. That's part of our capital day, which culminates in the talk by Professor haplotypes from the University. It's going to be a really cool talk about ants, the ultimate superorganism. But today the star is our own Harsha Barden, HT, as he's often referred to. Harsha came to us almost as an academic grandson, doing his undergraduate and master's degree at NG Prasad at ISO Mahali. Prasad was Joy and I, it's our first postdoc, we had a postdoc together 20 years ago, NG Prasad. And now we have the next in line, Harsha. I know Harsha especially well because 4 years ago on one day, 4 years and 1 day ago, of course, was the ominous poster day, Friday the 13th, a true Friday the 13th, where everything shut down and many people locked up their labs. Being the fly ranching side of the co-supervisor relationship, we had to decide a few really important things. Obviously, we had to keep our main stocks going. We also had a selection experiment, not just any selection experiment. The selection experiment that involved collecting 4,500 virgin females every 2 weeks. And that's something that HT and I decided to press on with. And so that means we spent a lot of hours here tending to that experiment, working together. I endured hours and hours of podcasts on socialist unionist history. Cricket matches, what else? What else did I ever put up with? We worked together, shoulder to shoulder in the lab. We kept that place going. I know him to be a hardworking individual, an exceptionally clever individual, an exceptionally good-natured individual, an all-around great guy, so generous at this time. I think anyone in the graduate student community knows of something that's partially help with something, whether it's taking a pet to the vet or helping with an emotional crisis or whatever it's been. He has worn many hats in our department, not just an outstanding graduate student, but he has worked for the department in terms of improving mental health, equity, diversity, and inclusion, inclusiveness issues. He has worked for the grad students as a bond with the union from improved conditions such as funding to grad students. He's again done a lot of things for a lot of people in this department, probably one of the most visible faces of the department. It's bittersweet then to see the end coming for Harsha. He's moving on to a fantastic postdoc fellow, Thomas Flack in Switzerland in September. So he's got the fire lit underneath him now to do this next seminar and do that defense and get on to a great postdoc. So welcome, Harsha.
Thank you so much. That was literally so kind. It's going to be hard to follow that introduction to the talk now. So thank you all for attending as well. I'm here to talk to you about intralocus sexual conflict and how we studied it with something called a male-limited selection experiment. So I'd like to get started at a very classical place for a lot of evolution talks, which is, why do we have genetic variance for fitness? The question follows from a simple thought experiment. You can imagine an illustration like this for some trait, let's say body size perhaps. The trait has a certain amount of variance in a population that's given to you by this little dotted line that goes across. And a subset of that is heritable genetic variation that gets passed down from parent to offspring. Now, let's say that this population has a fitness function that looks like this red line up here, and the highest fitness individuals correspond to about here trait values. I should be using this pointer, I have a pointer. And so right here, you would imagine that over time, natural selection weeds out the low fitness variance and you kind of move the trait values towards that section of the fitness function, but you also collapse or you erode genetic variation as you do that. So to put it in an analogy, if natural selection is the engine that gets traits from point A to point B in trait space, heritable trait variation is the fuel that it burns to do that. So why is there always fuel left in the tank? Why do we have heritable genetic variation for fitness? Now, it's not like we don't have any answers to this question. We know of a mutation selection balance. Mutations bring in variation, selection erodes it, and populations equilibrate at some kind of balance between the two. You have polygenic selection where traits have multiple loci that contribute to the trait value, and perhaps different combinations of alleles make out to a similar trait value that's being selected like this. And then you have balancing selection, which is this entirely interesting field of any form of disruptive selection that maintains polymorphisms within a population. So any kind of system where you have a trait distribution that looks something like this, where you have a peak over here and a peak over here. You can think about spatiotemporal selection gradients, like clines, perhaps. Maybe you have a population distributed across a mountain and animals higher up are being selected for different heat shock proteins than the ones below. Or you might have frequency-dependent selection, like with sexual selection, where unique males tend to be really attractive, but only as long as they remain unique. An additional contribution that we think could be playing a role is sexual conflict. So in sexually reproductive populations, you can imagine that much like a genotype by environment kind of interaction, where genes have different fitnesses in different environments. A gene might have a certain fitness value in a male and a different fitness value in a female, and variation of this nature might maintain polymorphisms within the population. So is there any reason for something like this to be going on? The sexes of the sexually reproductive populations tend to be anisogamous. This means that they produce very different gametes. These come in two common strategies. One is large, energetically costly. It tends to be expensive to produce and not very common. The other one is extremely abundant, cheap, and energetically very easy to produce. I'm talking about the egg and the sperm. Now, the downstream energetic economics of these two different types of gametes result in completely different selection functions or fitness functions for the males and the females, frequently causing them to be divergent in their interests over life history, ecology, behavior, and so on. However, in spite of these extremely divergent selection pressures, the sexes share a genome. A majority of the traits expressed by the sexes have extremely positive genetic correlations, which prevents them from evolving dimorphically in response to these divergent selection pressures. Now, this imposes a fitness load upon these populations, and we term that as intralocus sexual conflict. This is epitomized by a single allele having different fitness outputs in the males and females, as you can see in this landmark example here. So Susan Mills and team working in Finland in 2012 published a study where they are interested in testosterone, male testosterone in bank walls, and whether it underpins intralocus sexual conflict in these populations. So what they predict is that if that endocrine pathway is actually mediating sexual conflict, you would expect this kind of genotype by sex interaction coming out of different levels of testosterone in the animals. So they create artificial selection lines that have extremely low male testosterone and lines that have extremely high male testosterone. And they look at the fitness of the males and females coming from those lines. And here you can see that genotype by sex interaction. In the black, you have high fitness males and low fitness females coming from lines selected for testosterone. And in the white, you have the opposite pattern, low fitness males and high fitness females in lines selected against male testosterone. So, besides being interested in whether intralocus sexual conflict acts as some kind of capacitor for genetic variance, we were also motivated by a couple of other theoretical questions in this project. One of those is where the sexually antagonistic loci are within the genome. And so before I talk about the theoretical predictions here, I want to orient you to this little diagram. This is just a karyotype diagram of a Drosophila melanogaster genome. You have the sex chromosomes, the X and the Y, a pair of autosomes called the 2 and 3, which are substantial, and a dot 4th chromosome that I'll largely be ignoring for the purpose of this talk. Now, the X chromosome is haplodiploid. There's only one copy in the males, there's 2 copies in the females, and therefore you can accumulate male benefit, female deleterious variation on the X chromosome. As long as it's recessive, it's not going to be exposed to selection up until it reaches a certain frequency within the population. In a situation like this, you would imagine that these sexually antagonistic loci marked by these bars would be non-randomly enriched on the X chromosome and not so common on the autosomes. However, starting from an alternate set of assumptions, you can result in very different theoretical expectations. So if we believe that dominance of alleles depend on the sex in which they're being expressed, for instance, say a male benefit allele is dominant in males but recessive in females, then this haplodiploid X chromosome is no longer privileged over any of the other chromosomes, and you would expect this kind of more random distribution of sexually antagonistic loci across the genome. So with all these questions in mind, how do we go about addressing these problems? Ideally, you just want to shift from a sexually reproductive system to an asexually reproductive system and see what happens when you get rid of sexual conflict. Problem with that, though, is that you're getting rid of much more than sexual conflict. You're getting rid of systems that undergo recombination and break up linkage groups on a frequent basis. And so we're looking at extremely different evolutionary dynamics, and it's very hard to analyze these predictions there. So to explain how we actually get this done, I'm going to take you through a little detour about Intralocus fishes. Now, these fishes were all the craze in the 60s, late 60s and the early 70s, starting with this discovery by R.J. Schultz here in 1967. So he discovers this system of gynogenetic Intralocus fishes. And so these are hybrids, they're allotriploid hybrids, which means that they're 3 N genomes, each of those 1 N is coming from a specific population. And the PMLL here means that they are Monaca lucida lucida, which talks about each of their 3 genomes. Now, the Monaca lucida lucida females and PMLL females reproduce clonally. They produce MLL eggs, which require a sperm from the lucida population to trigger embryogenesis, but they don't actually incorporate any genetic material from there. So generation after generation, they're passing down just MLL genes from mother to daughter, and so on. And with this crazy system coming up, people were interested in seeing whether the other Intralocus hybrids were doing something weird as well. And that brings us to Mike Cimino in 1972, who discovers the hybridogenetic reproduction of P. Monaca lucida. Now, these females here are doing something even more interesting. They produce eggs that always contain only the M genome, incorporate a lucida genome from lucida fathers, and then produce an identical, well, not an identical, but a hemiclonal female that is exactly the same as the mother for the Monaca genome and a completely new lucida genome. And the way this happens is that during their weird meiotic process, they have unipolar spindle formation, where all the Monaca genes are attached to the unipolar spindles and taken into the gamete, while all of the lucida genes are just resorbed into the cell wall. And so what you have here is this really weird system, which is, while asexual, you have an awkward situation of sexual selection where these females are able to co-evolve with the males they mate with, but those males hit a dead end every generation. So rinse and repeat, every generation you have the Monaca genomes being passed down, the Monaca hemiclones being passed down, and the lucida hitting a dead end. Now, Drosophila is an amazing model organism. You've heard this a lot of times. You've heard it because it has a short generation time. It has a well-annotated genome. It's really easy to handle in the lab, so on and so forth. I'd like to argue that it's an amazing model organism for this study because of a very niche property of it, which is that Drosophila male gametes do not undergo molecular recombination. So there's no crossing over happening with the Drosophila males. The only gene shuffling that ever happens is the independent assortment of entire chromosomes. And if we are able to prevent the independent assortment of these entire chromosomes, we're able to recreate this weird system that the P. Monaca lucida is going. And before I show you how, I'd like to orient you to what I'm doing over here. Now, this is the karyotype from before. I've just dropped the 4th chromosome that I told you I'd be ignoring. But I'm representing it through these little boxes here. So an XY has a box with an X and a Y. There's a couple of 2s, there's a couple of 3s. That's what's going on when I put these diagrams up. When I mark things with color, they're generally being marked as pieces of interest to me. And you'll see a few other markings like this, which are typically like visible phenotypic markers that we introduce, some other mutations that we have value for. Now, in this system here, if I have a hemiclonal male with a Y23 haplotype of interest to me, with these recessively marked background genes and females that have the exact same thing, I can successfully pass down this Y23 from generation to generation while manually removing all the other variants based on visible phenotypes. So just by introducing markers, I can pretty much recreate the Monaca lucida system. I can take this one step further. I can introduce large-scale translocations between the autosomes, which introduce some aneuploid mortality and make the process a little more automatic. But that's just a little thing. Yeah. I also told you that I was interested in X chromosomes. So I can take this one step further and use females carrying an XXY karyotype. And these females over here, they have conjoined X chromosomes that can't be inherited separately. So anytime they're producing a daughter, they're producing a conjoined XX daughter. But anytime they produce a son, you're getting an X chromosome from the father instead. And now with this system, I can take the Drosophila male's capacity to shut down molecular recombination and pass down an entire X23 haplotype from father to son, generation after generation. I've been working with the system for a few years now, as Adam's mentioned, and one thing I frequently forget to mention is that this is really wild and cool. It's a very whack system. It's not common that we have these kinds of manipulative powers, but we have it here. So this is exactly what Bill Rice does in 1996, where he's the one who comes up with this system, working with famous postdoctoral researcher Adam Chippendale. He creates a replication of the Monarcha lucida system, where you can pass down X23 haplotypes from generation to generation with an egg from this weirdly marked female called a clone generator female. And then her genes basically hit a dead end, and these X23 haplotypes can evolve against these arrested females. And so that's exactly what he's interested in. He's interested in female coevolution arrest. He wants to know what happens to the dynamics of sexual selection when only one sex can adapt to the other while the other one is held in arrest. And what does he find? So over here in these plots, all of these white measures are control population measures, and all of the black ones are from selected populations from a selection experiment. He finds that the fitness of the selected animals are consistently higher across a bunch of different measures, suggesting that this capacity to adapt to arrested females actually improves the male's capacity to mate with them specifically. And so, yeah, that's the finding, increased fitness measures for the selected males. What he doesn't notice immediately, but he kind of gets around to a couple of years later, is that he's introduced a system of male-limited evolution. Now, Rice is still interested in his populations evolving rapidly. He doesn't want them to be entirely asexual. So using an ingenious system of pulling out these X23 haplotypes and putting them in females where they can recombine with each other, he's able to enable recombination in the system while restricting evolution to one sex. And so what he's created here is a system of male-limited evolution. And this system doesn't get explicitly tested for what it's doing, which is that it should be improving male fitness, but wherever you have sexually antagonistic polymorphisms, it's going to cost the females and benefit the males. This then gets tested by N.G. Prasad in 2007, working here at Queen's University. This is also where I started to get fascinated with all of these systems. But what they find is that in the selected populations, the square boxes compared to the controls in the circles, you have low female fitness in the male-limited populations and higher male fitness, and the opposite for the controls, high female fitness and low female fitness. So what they found was they were able to select populations to be male-limited, and they found increased male fitness at the cost to female fitness for all these selected haplotypes. So what do we have here? We have a system that promises to take a sexually reproductive species with some kind of trait distribution for the females, some kind of trait distribution for the males, different fitness functions across whatever traits, because we're just selecting them overall for their fitness. They are held together by intersex genetic correlations and pulled apart by the fitness functions of the two sexes. When we eliminate selection for the females, we should masculinize traits. So wherever polymorphisms exist in sexually antagonistic loci, they should be completely masculinized. Fitness should increase for the males, and the genetic variance should kind of be reduced because we're getting rid of a system of balanced selection. So explicitly, these are our predictions. We predict an increased male fitness, reduced female fitness, and we also predict a reduced heritable variation for male fitness. Again, this is quite landmark in the sense that nobody's very capably shown this. As intuitive as the verbal argument is that removing balancing selection should reduce heritable variation, we've never actually been able to test sexual conflict in this way. So we thought we had the exact system to do that. So what we did was we created some selected populations and controls. We picked 3 populations. They're called C135 because they have a history in this lab. We didn't call them the 1, 2, 3 because we didn't create them. And then we derived from each of these a pair of populations, a matched control or an MC, and a male-limited population or an ML. Now, these colors that you see here, they're going to stay consistent across the presentation. So whenever you see a turquoise blue, I'm talking about something male-limited, and an orange is matched control. And As I mentioned earlier, the male-limited populations are hemiclones that are evolving in this male-limited fashion. So you'll notice that their karyotypes also look kind of different. The male-limited populations are heterokaryotypes, where they have a haplotype of interest to us and a bunch of genes from a clone generator female, whereas the match control animals are completely from a control stock baseline. There we go. This work has been happening in the lab for in excess of 90 generations. Every week, for about 6 days, between day 9 and day 14 of these animals' life history, our lab day kind of gets dominated by the maintenance of these animals, the maintenance of the many different stocks that are required to propagate these animals, and the maintenance and creation of all the stocks that we need to do, that we need for breeding designs for subsequent experimentation. This work would not have been possible without these 4 people here. As Adam mentioned, this was a pandemic project. I was the only person in the lab at that point of time. Adam rolled back his sleeves and began working in the lab and continued working after the lockdown. And besides all of his conceptual support, this wouldn't have been possible without his labor. I'm deeply appreciative for that. At multiple different points, I've depended heavily on Imran, Josh, and Mindy to keep this project alive. And I'm also deeply, deeply appreciative to them for their support. Okay. So we do all of this. As I mentioned earlier, we have extremely different looking karyotypes. We have these heterokaryotypes for the male-limited animals, and these match control animals look like homokaryotypes. We don't want to test them under these conditions. And the reason we don't want to do that is because we're not really interested in the fitness output of these clone generator haplotypes. So we come up with a breeding design that gets rid of these and expresses the MLs, again, as heterokaryotypes, but with a haplotype coming from the matched control populations that's paired with them, rather than from a completely random pack. And so occasionally, while I refer to this, you'll hear me use the words wild type. All I mean is that we're getting rid of all of these weird hemiclonal genetics here. Other things that I'll be referring to, I'll be talking about replicate populations. Those refer to population pairs 1, 3, and 5, and selection regimes as NC and ML, as I already introduced them to you. Another thing that we require is recessive competitors. The reason we need this is that for assays like competitive fitness, we need some kind of animal that we're comparing, some kind of baseline, and we have phenotypically marked recessive animals that kind of get this job done for us. Okay. So prediction 1 was that we would see increased male fitness at the cost of female fitness. How do we go about assaying reproductive fitness? We try our best to mimic the evolutionary conditions of these animals. So they're combined, the males and the females are combined between days 12 to 14 in large cages with large population sizes. We don't go exactly that far, but we create these little collectors where 30 target animals compete with 30 competitors to mate with 50 animals of the opposite sex. They stay together for day 12 through 14. We collect eggs from them, and then we visually phenotype the progeny that's being sired or dammed by our target animals. Once we do that, we have estimates of the trait, and which is fitness in this case, and we also have estimates of trait variation. What we don't have is estimates of heritable trait variation, for which we go back to our hemiclonal system. Now, because we're able to reliably passage an entire haplotype and put it wherever we want, put it in a male background, put it in a female background, and so on, we're able to create entire lines with half their genomes being exactly identical and another half being randomized samples from a control population. And so we create 40 such ML hemiclonal lines and 40 such matched control hemiclonal lines with the support of these clone generated variants. So what did we find? Going backwards in terms of our results, our hemiclonal fitness analysis gave us a couple of really interesting findings. So what I'm showing you here, the histograms in the background are our actual measured estimates of fitness. The dotted lines that are going over here are estimates of trait variance. And the subset, the inset block distributions are the heritable trait variation associated with these populations. So you'll see here already that the MCs in red or orange are a little thicker, the MLs are a little shorter and a little taller and a little shifted to the right. So as we predicted, the fitness of our selected males increased by 21%. This was extremely significant. And the genetic variance for fitness reduced by about a factor of 3. Again, extremely significant. And we thought that we cracked it. We come up with the first empirical demonstration that sexual conflict was maintaining genetic variance. We thought we had a little nature communications situation going on here. But before I get any further, I also want to tell you about the intersex genetic correlation for fitness. I didn't outline any predictions about this because it's a bit of a wonky concept, the idea is that if you have a negative intersex genetic correlation for fitness, so say you have high fitness males who are also low fitness females, and low fitness males who are also high fitness females, you would have a kind of negative slope running through here. And that negative intersex genetic correlation would be a really good diagnostic tool of intralocus sexual conflict in these populations. So perhaps we would have seen something like the matched controls have a really negative slope, and then the male limited populations kind of equilibrate. The trouble is that this diagnostic tool breaks down when you have a zero or near zero genetic correlation for fitness. And the reason is that populations can accumulate sexually antagonistic and sexually concordant variation, which kind of averages out. And in that case, you simply don't know if there was any intralocus sexual conflict. Both of our populations showed no different a slope from zero, correlations that were roughly zero. But here as well, you can see that the male limited populations have had higher male fitness than their matched controls. Okay. And now the trouble. So we had independently done a bunch of competitive reproductive fitness assays. In panels A and B here, I'm showing you male fitness from different generations. And in panels C and D, I'm showing you female fitness. As you can see here, the male fitness of these populations didn't look any different from their matched controls. Across multiple assays, we found no significant differences and barely any effect size between them. But female fitness, on the other hand, did reduce in the male limited populations as we predicted relative to matched control populations. So taken together, we were a little confused at this point. Prediction 1, which was our big proof of principle that the male fitness of male limited animals would increase, wasn't quite working out. However, prediction 2, that reduced genetic variance would be seen for male fitness in the male limited populations, was coming through. And there was this giant contradiction where the fitness data from the hemiclonal analysis and our competitive reproductive fitness assays were not showing the same patterns. We started thinking about this. We were wondering if we picked baseline populations that didn't have enough standing genetic variation to do the job. Perhaps we had ML haplotypes that were completely recessive to the controls that we paired them with. Or perhaps there were some alternative sources of selection that were producing local adaptation to selection conditions unrelated to intralocus sexual conflict. Second here. And so this contradiction in our two assays was actually a guiding light, helping us figure out what was going on here. So in needing to produce exactly the same haplotype in male backgrounds and female backgrounds and create these hemiclonal lines as such, we had to accommodate a slightly compromised breeding design that didn't make a completely wild type animal in our hemiclonal assay for fitness. So as you can see here, this has an X23 haplotype from a male limited background, and it has the 2-3 chromosomes from our randomized control background. But we were forced to incorporate a Y chromosome that came from our clone generator females to make this design possible. On the other hand, when we were testing competitive reproductive fitness, we'd gone much further and created a proper breeding design that ensured a proper heterokaryote between the male limited and matched control populations. So immediately, we started thinking that this Y chromosome here was perhaps contributing to these fitness differences that we were seeing. And so we started thinking about the alternative sources of selection that could be contributing in this selection experiment. So the very first piece is one that we're already familiar with. We know of female coevolution arrest. This is what Bill Rice was initially interested in. We're not particularly interested in it, but it's possibly creating a response. And if it dominates the response, then maybe you aren't seeing a response in other places. And then within the genetic background, there's a bunch of different factors that we have to account for that's traditionally not being given much consideration to these studies. The first of those is a novel cytotype. When you expose males to completely new cytoplasms, which include mitochondrial genetics, you expect certain fitness effects that I'll talk about in more detail in a bit. So there's that to deal with. There's these weird translocated autosomes that we introduce to create some automatic culling and reduce our effort. And there's the shared Y chromosome that splits between a male and a female in every generation, unlike any other Y chromosome, which just goes patrilinearly. So there were multiple other things that we took into consideration with this genetic background, but I'm going to focus on these three for today's presentation. So what we realized very quickly was that besides these two original variables that we were interested in, replicate and selection, we had a couple of variables that were completely collinear with our selection treatments. The first were those with the females. So the matched controls were mating with control identity females, and the male limited populations were mating with the clone genetic females. The second was the genetic background. The matched control populations were under what I'm calling a wild type background, and the male limited animals spent all of their times in these weird genetic backgrounds, which I'm referring to as home. So we decided that we would go back to the drawing board, draft breeding designs that allowed us to do a full factorial assay that allowed us to pitch all of these variables against each other completely and see where those kind of selective or adaptive responses were coming. Okay, this is a big messy graph. The first thing you'll notice is that I've gotten rid of the replicates. I've collapsed all the replicates into one, and that's because we saw no replicate effect or any interaction with replicate. What are the big results that we saw here? So first, let me mention that we saw major interaction terms, significant interaction terms between selection and background and female and background. So when we kind of take all of the pairwise combinations of selection, background, and female independently, what do we see? The first thing that we notice is that selected haplotypes perform better than controls against both kinds of females in the home coat background. So you can see here in home coat backgrounds here, the male limited animals in blue perform better than the match control animals, with both the clone generator females and the recessive conflict females. So clearly there's something going on in the genetic background that these males are adapting to, which is unrelated to intralocus sexual. The second thing that we realized was that these selected haplotypes perform no better than controls tested against both females in the wild type background. So absolutely no trend here. A bit of a trend here that was bordering on significant, but it didn't achieve the alpha threshold that we had. And so what we were seeing here, if you kind of average over these two plots, so the home coat animals, the slope that they display going from match control to male limited with both females relative to the same that's happening in wild type, you see the basis for our first interaction already. So as you go from match control to male limited, you have a greater slope in home coat animals and a lesser slope in wild type animals. So that's where our first interaction was coming from. And then where was the second interaction from? So we looked at the two genetic backgrounds, fitness going from the home coat to the wild type with both sets of females. You can see here that under the wild type background with the recessive competitor females, where all the artifacts have been stripped away, and that should be our generic fitness test, our animals didn't see the fitness benefit that we were hoping for. And so this slope is drastically reduced compared to the same that we see with clone generator females, where there's that nearly significant trend of difference. And from there, we get our second trend, which is our second interaction, which is that as you go from home coat to wild type, you have different slopes with clone generator and recessive competitor animals. This was a bit of a blow. We didn't have our Nature Communications paper because the genetic variance was being reduced for reasons perhaps unrelated to intralocus sexual conflict. I want to take pains to mention that this doesn't mean that there was no intralocus sexual conflict in these populations. The adaptive responses we were seeing were coming from elsewhere, and they were so dominating that we weren't able to identify what was going on in terms of intralocus sexual conflict. So what next? We decided we would figure out where the fitness components were coming from. Were these animals different in the levels of mating success they were achieving? Were they different in terms of the way they were interacting in terms of sperm and so on? So to figure out whether they had different mating success, we would put pairs of males, a target and a competitor male, together with a female. And then we would wait for one of them to mate. We would record the amount of time it took for them to start mating, the amount of time it took for them to mate. We would release the unmated male, and we would record the identity of the mated male to figure out mating success. And we would phenotype the progeny to confirm the ID of the mated male. We would count the number of animals they produced to see how much fecundity these males were able to initiate. And we would also look at their brood sex ratios, because our weird sex chromosome system gives them an opportunity to have some kind of drive system evolve if the variation for it exists. Then we were interested in sperm competition. The arrest to a very specific scenario of sexual selection was perhaps inducing selection for males to develop their sperm in certain ways, or perhaps the seminal proteins that went to the sperm and so on and so forth. So we were interested in sperm organs, and to do this, what we were doing was we were combining the females that we were using with one competitor male first, allowing them to get mated, remove the male, allow them to process and manipulate that sperm overnight, and then combine them with the target male after, and 24 hours later, we would collect eggs from these females, and we would figure out what proportion of progeny our target males had sired. We were also interested in whether these males were modulating the levels of mate harm they were putting into it. And the reason for this is that these clone generator females we use with all these weird mutations, they tend to be extremely fragile. They are extremely vulnerable to death from mating, And therefore, we were interested in seeing whether these males approached this any differently. Perhaps they were evolving to be less harmful and indirectly boosting their own fitness. And so to do this, we would combine the males and the females for 2 days between days 12 and 14, as they usually are. And then we monitored mortality starting with day 12 all the way to day 19, and looked at the pupil productivity from the females that were in these vials. Okay, so what did we find? Lots of things going on in this slide here. I'd like you to focus on this corner first, where I'm talking about mating success. So quite consistently, we found that our selected haplotypes were more successful at mating. They were getting a much higher proportion of mates than their matched controls. And this seemed to be true across pretty much all the conditions that we were testing them in. We associated with this, we also, as I mentioned, tested for fecundity initiation, brood sex ratios, mating duration, and so on. We found no differences in any of those across any of our variables. Mating latency did reduce. So it seemed like our male-limited animals were much quicker to access a mate compared to the matched controls. Looking at sperm offense, we saw effects of the females that we were using. We saw effects of the backgrounds that these animals were in, but we saw no effect of selection or any interaction with selection, suggesting that these animals weren't improving as sperm competence. And then we went on to look at mate harm, where we found that under both genetic backgrounds, our animals were less harmful to these clone generator females they were mating with. And under a wild-type background, they were also able to use this to indirectly boost their own productivity, perhaps boosting their fitness, those experiments we were discussing earlier. Okay, and so we teased apart where the fitness components were, what was going on there. And then we were interested in teasing apart the genetic background and what aspects were actually driving these adaptive responses. So what were the major concerns we were dealing with? The first was translocated autosomes. I've already told you that we use very weird translocated autosomes, and there might be a mutation load that's associated with this that's non-visible, but might be affecting the fitness of these animals. Because the male-limited animals are the only ones exposed to them, there might be adaptation that's going in that direction rather than towards the resolution of intralocus sexual conflict. The second thing we were concerned about was mother's curves. Now, I already mentioned that exposing a male to a novel cytoplasmic background causes weird fitness outcomes. And the reason for that is the cytoplasm is matrilinear in its transmission. So it goes from mother to daughter and son, but only from that daughter subsequently on to the next generation. So you can imagine a situation where mutations accumulate in the cytoplasm or the mitochondrial DNA, for instance, where it is agnostic to male fitness, perhaps causing fitness costs as well. And any adaptation to counter those costs that are being experienced by the males has to happen in the nuclear genome of the male through some form of nuclear recovery, which is all well and good as long as these populations remain in tight interaction. But if you are far removed from the animal whose cytoplasm you're receiving, you might be exposed to all of those costs all of a sudden. And so, given that that's exactly what happens in the male-limited selection experiment, we were concerned that the cytoplasm might be introducing cost. And then thirdly, we have these female-exposed Y chromosomes. Now, Y chromosomes, just like the cytoplasm, are also sex-limited. They go through the males alone every generation in their transmission. And they tend to be critical players in the regulatory scheme. So a Y chromosome, although extremely degenerate and gene poor, is going to be playing a role in regulating other chromosomes within the male genome. And these weird Y chromosomes that we use in the experiment spend their time 50% of the time in the males, 50% of the time in females. So one of two things could be happening. Either they're not undergoing selection in the females, but they're undergoing relaxed selection, accumulating mutations 50% of the time, or they're undergoing selection towards female fitness function, which would be extremely concerning for what we're doing here. Other studies with different XXY karyotype melanogaster flies actually show that these Y chromosomes seem to be regulating mating courtship behaviors, which would be of huge concern to us given what we're interested in. So I've already told you that we have these weird breeding designs where we were able to introduce just the Y chromosome from a clone generator animal and nothing else. Similarly, we were also able to create a breeding design where we were introducing just the translocated autosomes and nothing else. And Adam fortuitously slipped upon a cytoplasmic mutant where we were able to separate the conjoint X, which hasn't happened in a really long time, and we were able to introduce that cytoplasm as well. So essentially, we were able to tease apart all of these three alternative sources of selection and test responses in the male-limited animals and their matched controls. What we found was that we had a consistent sign of adaptation to the CG cytoplasm, the clone generator cytoplasm, with male-limited animals being significantly better in all replicates. In 2 out of 3 replicates, animals with a CG Y chromosome were better in the male-limited selection compared to matched controls. And we were able to pull out no effect with the autosomes, but we saw a trend approaching significance in just 1 of the 3 replicate populations. So what this tells us is that even before we account for all the possible interactions between these different alternative sources of selection, we're already seeing fitness effects. And so there's very clear signs of local adaptation to all of these genetic oddities that we have introduced to make this system possible. So what have I told you today? I have told you that we have evidence for adaptation to alternative sources of selection, which obscures our ability to study intralocus sexual conflict. Selected animals seem to be adapting to both the normal genetic background and the clone generator females that we paired them up with. The improved fitness is being mediated through mating success and reduced mate harm, not so much by sperm competition. And we have evidence for adaptation to the cytotype and the Y chromosome, but isn't identical across all replicates. What are the major takeaways from everything I've told you today? Future experimentation with these systems, the male-limited system or the hemiclone system, requires much more rigorous controls. Past experiments that have depended on these have not really taken into account a lot of these things. And so we might have on our hands some spurious findings that we need to account for better. Additionally, there need to be more base populations that go in to study the generality of these findings. Adam's already told you how I'm an academic grandson. This niche of the field is extremely inbred. It's a bunch of people working on the same populations over and over again. Every study, barring 2, that has used these systems, which is about 20 of them so far, have all used flies from Larry Harshman's lab called the LH moderates. And they all seem to be panning out in a similar direction, but what do we know about this being general to even Drosophila melanogaster before we push these findings anywhere further? So our study represents a first or a third, perhaps, of doing this with a different population and trying to figure out what's going on, and we're not able to replicate what has been seen in the past. And so with all of that, I would like to stop here and acknowledge people and situations that bring me here today. So my place of work is situated on stolen land. Today, this territory is included in the Dish with One Spoon Wampum Belt Covenant, an agreement between Haudenosaunee and Anishinaabe nations to peaceably share and take care of the resources of the Great Lakes. Newcomers were later invited into this agreement in the spirit of friendship and respect. Second, I work on a land very far from home, in search of improved economies and the research infrastructure that they bring. These improved economies themselves are a product of imperialist accumulation in the global south, and the enforced terms of a neoliberal economy or a neoliberal consensus that continues to fail the median global citizen while enriching the owners of our economies. So when Canada scapegoats international students and workers and limits their access to these resources in the name of fairness, this is the historical context for it. It's a wretched distortion of logic facilitated by an appeal to common sense. Within an Indian context, the limited access to these resources that I'm talking about here are further unevenly distributed. This may not mean very much to you, but I'm a member of an oppressor caste within the Hindu religion. And I've had an extremely privileged access, a privileged life in many ways, but a privileged access to education and the additional resources that brought me here through an accident of birth. Okay. Having acknowledged the structures that situate me here, I want to thank the people I've worked with. I've spent my time here overlapping with Dr. Josh Alpern and Imran Syed, our department's newest PhD candidate. These are 2 kind humans and brilliant minds who I've learned a lot from, and I'm extremely grateful for the work environment they've fostered and the friendships we've shared. During the time I've worked on my thesis, I've had the pleasure of mentoring 8 honors thesis students and their research projects. Tomorrow, if you're attending the poster session Adam talked about, you'll get to hear from Josh Kowal and Caitlin Vio. Josh is describing morphology data and life history data from these selection experiments, and Caitlin will be talking to you about correlated female responses to the kinds of male adaptation that we've seen in this experiment. The 2 of them, along with Avery Want and Mindy Baruti from the class of 23, have contributed immensely to this project and will feature as co-authors on various manuscripts related to this project. Mindy is also a remarkable artist. All of the cartoons that you're seeing, this one included, is Mindy's work. She is available for hire as a scientific illustrator. You can find her on Facebook or reach out to me, and I'm happy to put you in touch at any point in time. I've also worked with Emily Mendelson, Katie Faulkner, Amanda Wigney, Nav Navamenan, and Krista Robertson. These 5 students performed incredibly in the lab, but also worked through a very fraught time where their theses were disrupted by the pandemic. But I really treasure the time working with them. And last but not least, Chloe Robinson was the very first thesis student I worked with. And I'm really proud to share authorship with her on my first publication. I'm grateful to Adam and Troy for their guidance and supervision throughout this project. A good supervisory relationship is honestly half the PhD done. And I really can't take what we have there for granted. Which also reminds me, all of this work was made possible by NSERC grants to Adam and Troy. I'm thankful to Paul Martin, Janice Friedman, and Maria Aristizabal for their work as committee members. My project has pivoted multiple times, once for the pandemic and a couple of times because of really weird results we had. And they've stuck with me through all the twists, giving me extremely useful guidance all the way through. And of course, my chosen family, friends back home who are attending the Zoom call in the middle of the night. Friends in the broader Kingston community, friends in this department here, this would not have been possible without you. Previous graduate students in this department have created a beautiful culture with leaving us with a tiny piece of unsolicited advice on their way out at the exit seminar. I'd like to carry the baton with this, except instead of addressing my unsolicited advice to graduate students who come behind me, I want to speak to the department at large. The funding system that we're dealing with is not a sustainable situation. I know that supervisor grants are being maxed out. I have seen biology put in the efforts to stretch a tight budget. And even with this, the grads are in really poor financial health. In our unfunded years, which it seems like everybody is going through, this is especially harrowing. We need to stand together to demand better from this university, from this province. At the end of the day, a university is a public institution. To treat it as a business is a disservice, but to mismanage it at these historical proportions is a grave insult. And with that, I'll take questions. Yes, go ahead.
So that was an excellent talk, thank you. And I think despite being on your committee and some of Adam's other students' committees, I think I finally understand the heritable system. Don't look at me like that, Troy. So you ended by suggesting that, there's sort of problems with using this genetic design that you guys do, and that future experiments need better controls. And I'm wondering if you have suggestions. I mean, I know like the thing you showed at the end, trying to get at the cytoplasmic effects, like is that what you're anticipating would need to be done from the start, or how, I guess, how would you have done it different, the first experiment different?
No, this is really valuable. And this is one of the pieces that Adam and I are like working on writing together right now in terms of how you would go about this. It's featuring in one of our manuscripts. So hemiclonal designs have been used to do 2 different kinds of things. One is assay heritable trait variation, and the other one is to create these kinds of funky evolution experiments. As far as assaying trait variation goes, we can control this pretty well. So the heritable variation assay that I showed you controls for everything except that funky Y chromosome. And we can go one step better there, because if we knew that we had a Y chromosome issue, we would simply back cross a different Y chromosome into those populations 1 generation earlier. It's a very easy thing to do because the Y chromosome is also so easy to back cross. And if we'd achieved that, we would have perhaps been looking at results without artifact whatsoever. The challenge, of course, was we figured this out too late, and running a hemiclonal analysis takes months and months and months. And so I think that's an easy fix. There's an easy way to estimate heritable trait variations using these systems. Male-limited selection experiments are more challenging. And the reason is that even if you do backcross in a Y chromosome or whatever, you are still going to expose them to selection in this like male-female shared context, you're also going to not be able to avoid the cytoplasmic background which these males evolve with. And so one of the things that come to mind is creating the kind of genetic equivalent of a sham treatment. So like what happens if you have some male limited populations, but some other populations where you just introduce the novel cytotype alone, and then another population where you introduce just the Y chromosome alone. The trouble with that is that it exponentially increases the workload of an already very busy project. So it would be something that perhaps 3 people could like focus on for an entire thesis, but I don't know that it's a very practical suggestion. Yep.
I think I understand it now, but all so it is a problem, like, in a dream world, we kind of not anything with that, right? So in your experiment, it's problematic. The chromosome stack from the males that's always being transmitted, it's paired with a stack that's going to be combined from the clone generator female, right? So the assumption that if you're going to look at differences, that has been transmitted through all those generations, and you look at it has to have like a dominant fact. It's and that's why sometimes we didn't see differences because maybe it's recycling.
So are you?
And I think you mentioned it, but I'm wondering, this is a wild thing. If you can get, there are sex determination genes in Drosophila and Trop too.
Yep.
And there's like TF one where you can take an identical genome and it will develop a female and switch it to a different temperature. Like if you have a clone, switch it and it will develop a male, and then if you compare those, you do expect different ranges.
So what you're talking about is taking a haplotype and expressing it in a female and then identically in a male, and then looking for fitness consequences associated with that.
Yes, so they would have, the only thing that's changed really is the allele copy. They will have identical genomes, but the phenotype.
Yeah, and you could conceptualize a selection experiment very easily, where you do this generation after generation, and you have the only difference being the sex determinant. There's other similar work that's done through experimental manipulations rather than some kind of CRISPR-Cas9 system that would treat the genes. So there have been people who have put certain genes in linkage disequilibrium with the sex. And this is another Rice paper who's done a lot of the foundational work in this field. But the trouble, I think, with what you're talking about is that the reproduction then becomes entirely clonal. And if you have entirely clonal reproduction, you're back to asexual dynamics. And if you lose recombination, you lose the capacity to break up linkage groups, and therefore the dynamics of evolution will be fundamentally different. So I don't know exactly what you'd anticipate coming out of that, except perhaps we could hazard a guess that it would be a little more resembling an asexual system altogether, if that makes sense.
I mean,
Maybe I'm misunderstanding.
You just do what you're doing, you have your male selected line, and then you have female, so that's what you're comparing that. If you just switch those females to male, that gene thread, but how will those males differ from the males?
Right, and so what's happening here is that you have a population produce males and females as usual, but then you activate some kind of gene that then turns all the females into males as well, and you derive the males from elsewhere. Yeah, my initial concern is just about recombination, but I think there's a way to make that work where you have some kind of additional recombination system that accounts for the problems like we do. It could be a really clever system to address these exact same set of problems. Thank you. Yeah. Okay, anything on the Zoom?
No.
Okay. Awesome. Maybe we're all done. Thank you so much for coming out. Thank you.
Thanks for. Such.